Welcome to INDIGO Biosciences Assay Tutorials. This tutorial demonstrates INDIGO's 96-well pre-incubation format assay. Following the pre-incubation protocol, the cryopreserved reporter cells are thawed, then dispensed into assay wells and allowed to recover for approximately 4 hours before adding the test compound treatment media. The assay requires 2 days to complete. The assay workflow is depicted here in its entirety, but the following tutorial provides a detailed step-by-step description of the plate setup and processing. It's important to perform all day one manipulations of the assay plate inside a biosafety cabinet or laminar flow hood and to use proper sterile techniques. For greater visual clarity, however, the following demonstration is being performed on an open benchtop. The day one workflow begins with retrieving the assay kit from -80 degrees Celsius storage. This is a critical step because the reporter cells contained within the kit box are temperature sensitive. Use a sharp instrument to open the tough plastic bag, then carefully cut through the seal in the kit lid. Without delay, retrieve the tube of reporter cells from the kit box and submerge it in dry ice. Work quickly so that the tube of reporter cells does not warm up significantly above -80 degrees. Back at the lab bench, transfer the tube of cell recovery medium directly into a 37 degrees Celsius water bath. Transfer into a refrigerator the tubes of compound screening medium, detection substrate, detection reagent, and the vial of reference agonist. Retrieve the white 96-well assay plate and place it in the biosafety hood to equilibrate to room temperature. Next, use the warm cell recovery medium to perform a rapid thaw of the frozen reporter cells. This is a critical step. First, retrieve the cell recovery medium from the water bath and sanitize the outside surface with an ethanol swab and put it into the biosafety hood. Next, transfer the tube of frozen reporter cells out of the dry ice and into a rack in the biosafety hood. Without delay, dispense 19 milliliters of warm cell recovery medium directly into the tube of frozen cells. Immediately transfer the tube into the 37 degrees Celsius water bath for a minimum of 10 minutes. This allows for complete thawing of the frozen cell pellet. Now dispense the reporter cells into the assay plate. First, retrieve the tube of reporter cell suspension from the water bath and sanitize the outside surface of the tube. In the biosafety hood, gently invert the tube of reporter cells several times to ensure a homogeneous cell suspension. Pour the cell suspension into a reservoir and using an 8-channel pipette, dispense 200 microliters per well of the cell suspension into the assay plate. It is important to prevent cells from settling in the reservoir during the dispensing period. Transfer the assay plate into a cell culture incubator. The internal incubator environment should be 37 degrees Celsius, 5% CO2, and approximately 70% relative humidity. Allow the plate to incubate for approximately 4 hours. While the reporter cells are pre-incubating, use compound screening medium to prepare the various test compound and reference compound treatment media that will be applied to the reporter cells. Note that if you are intending to perform antagonist-mode assays, first supplement the compound screening medium with the reference agonist. We recommend adding a concentration that is equal to the EC80 value of the agonist. Retrieve the assay plate from the incubator and discard the media contents. The recommended method is to manually eject the media contents into a waste container, then gently tap the inverted plate onto a clean, absorbent paper towel to remove residual droplets. Cells will remain tightly adhered to well bottoms. Next, dispense into the appropriate assay wells 200 microliters of the prepared treatment media. Transfer the assay plate into a cell culture incubator. Allow the plate to incubate for 22 to 24 hours. It is now day 2. Today's manipulations do not require special regard for aseptic techniques and therefore may be performed on a benchtop. Approximately 30 minutes before intending to quantify receptor activity, remove detection substrate and detection buffer from the refrigerator and place them in a low light area so that they may equilibrate to room temperature. Turn on the plate reader and set it to luminescence mode. Program the instrument to perform a preliminary 5-second plate shake before reading the first assay well. Program the read time to 500 millisecond per well. After the tubes of detection buffer and detection substrate have equilibrated to room temperature, combine them to generate luciferase detection reagent. It's most convenient to perform this step directly in the media basin. Gently rock the basin several times to mix the solution, then set it aside for later use. Retrieve the assay plate from the incubator and discard the media contents. The recommended method is to manually eject the media contents into a waste container, then gently tap the inverted plate onto a clean, absorbent paper towel to remove residual droplets. Cells will remain tightly adhered to well bottoms. Dispense 100 microliters of luciferase detection reagent into each well of the assay plate. Allow the assay plate to rest at room temperature out of direct light exposure for 5 to 10 minutes. Transfer the assay plate to the plate reader and press Start to quantify luminescence per assay well. The raw RLU data generated by the plate reader must now be mathematically manipulated. Replicates RLU values are first averaged, and corresponding values of standard deviation are calculated. Averaged RLU values should then be normalized to allow for accurate and meaningful interpretations of the experimental results. For agonist assays, normalized data are most commonly presented in one of two ways, as depicted here. The left-hand plot presents data in terms of fold activation, also referred to as signal to background. The right-hand plot presents data in terms of relative % activation. Specifically, the activation response delivered by the test compound is divided by the maximal activation response of the reference agonist. This ratio is presented in terms of %, whereby the maximal activation response of the reference agonist is equal to 100%. In both examples, the normalized data are plotted against the respective log-transformed concentrations of the test compound treatments. For antagonist assays, it is particularly helpful to normalize averaged RLU values in terms of relative percent inhibition. Specifically, normalizing to the maximal inhibitory response of the reference antagonist, which has been mathematically manipulated to equal 100% inhibition. Again, the normalized data are plotted against the respective log-transformed concentrations of the test compound treatments. Thank you for watching this tutorial. If you have questions, please don't hesitate to contact Indigo's technical service team at the listed email address.